Dear candidate, dear Mr. Ying Gong, we're going to start in a minute, but we first give you the opportunity to get a little bit acquainted to the members of your corona. We do, as you can see, we have a full corona. And I noticed since you are in China, you still use the background of the UN defense opportunity, which is I compliment you. So first of all, welcome to all members of the corona, welcome to the promotion team, and most importantly, welcome Mr. Gong. I'm not saying Dr. Gong because that's going to be maybe in 1 hour from now. Hereby, I officially open this academic ceremony in which Mr. Ying Gong will defend his academic thesis entitled The Combination Integration of Redirect NK Cells for Cancer Immunotherapy. Mr. Gong, may I invite you to present a summary of your studies and the conclusions of your studies in the next 15 minutes, and I would like to give you the word.
Thank you, the introduction from Prorector. Dear Prorector, dear members of Corona, dear family, friends, audience, in the next 15 minutes, I will shortly introduce my PhD work, which is conducted in Professor Gerhard Boss in Maastricht University. So now I'm going to share my screen to you now. So my PhD thesis title is Combination and Integration to Redirect Natural Killer Cells for Cancer Immunotherapy. Here is the 2020 WHO statistic data. It shows there are more than 20 million new cancer cases were diagnosed in last year. That means cancer is still a malignant disease threatening human beings' healthy. And there are several therapy approaches to control the progression of cancer, like surgery resection, radiotherapy, chemotherapy, but the currently 3 methods have 2 obstacles. One is this method cannot distinguish healthy tissue and cancer tissue. That means these therapies always will damage some part of healthy tissues. And the second obstacle is when the cancer transferred in another place, we call them metastasis, especially in the central neuron system. These methods cannot get access. Thus, recently decades, one method is called immunotherapy, have overcome these 2 obstacles because immunotherapy was based on human immune systems, and it was very high specific and can get access to the central neuron systems. Based on the components, cancer immunotherapy can simply divide the 2 categories. One is protein, like cytokines, antibodies. They can boost the immune systems. And then the other category is called cells, like natural killer cells, T cells, and the dendritic cells. These cells are mainly derived from patients, and then will culture outside the human body, and then will activate it and expand it, then will transfusion back to patients. This whole process is called adoptive cell therapy. And my PhD work was related on natural killer cells, which also short for NK cells. So what is NK cells? NK cells is an important innate lymphocytes running in our human blood and also resident in our tissues. And in the cytoplasma, there's a lot of granules, which is the killing weapon of the NK cells. And on the cell surface, NK cells high express CD56 and the CD16 Fc receptor. On the one hand, NK cells express the activation receptors these activating receptors can recognize the ligands from the target cells, and they will transmit the signaling to secrete granules, kill the target cells. On the other hand, NK cells also express a lot of inhibitory receptors. They can, these receptors can recognize the ligands on the healthy cells, and they will deliver signaling that do not kill these healthy cells. So the activity of NK cells was sophisticated, regulated by the signaling from these two kinds of receptor. Currently, there are several clinical trials have demonstrated only NK cells solo therapy is not very effective. They need a lot of combinations or modified. So there are several concepts that can enhance the function of NK cells. One is based on anti-tumor antibodies. So the antibody can recognize the tumor antigen on the target cell surface, and then it will recruit the effector cells through the Fc receptor, and the NK cells will secrete the granules to kill the target cells. On the other hand, a second approach is we can give the NK cells more activation receptors. We can give more activation signal, then this NK cells is much more potent. And one recently technology was called the Chimeric antigen receptor. It is also based on this concept. I will introduce shortly. And the third concept is we can reduce the inhibitory signaling on NK cells either by a blockade antibody or use the gene knockout strategy. So we are now looking at the first concept, and we look at more detail on the antibody structure. So the antibody structure on the top, we call the FAB domain, which is the antigen binding domain. On the bottom is called FC domain, which can recognize the Fc receptor on effector cells. There are also some side chains to decorate the NK cell antibody. And here's the image how antibodies recognize tumor antigens and also recruit the NK cells to kill the target cells. And several groups have demonstrated if we remove the sugar chain on the Fc domain in an antibody, it will enhance the binding affinity of the Fc receptor on NK cells, and the NK cells will be more activated and secrete more granules to kill the target cells. So based on this concept, we have invented an antibody targeting mucin-1 antigen. And here we have a normal antibody, and also we include the control antibody, which target on the irrelevant antibody. And also we have a modified antibody that the sugar chain in the Fc domain has been removed. So when we conduct a NK cell killing assay, just add NK cells and tumor cells together, NK cells can kill about 50%. But when we add the irrelevant antibody, it will not influence because the irrelevant antibody cannot recognize the tumor antigen. If we add our normal antibody, it will increase the 15% killing capacity on NK. And when we add our modified antibody, it will further enhance that. But we want to reach 100% killing capacity of NK cells. So we want to generate a more potent NK cells. That's why we apply a recently technology. It's called the Chimeric antigen receptor. So this technology was based on basically transplant the antigen binding domain from an antibody and make an artificial receptor expressed on NK cells, and the receptor will express the antigen binding domain on the cell surface, and then there's a transmembrane domain to make it stable on the NK cells, And also, there was several intracellular activation signal to boost the function of NK cells. So we design these three parts into a lentivirus plasmid vector. And also we include the detection marker like a GFP and ING alpha. And then we package this lentivirus plasmid into a lentivirus particle. And then we simply add the lentivirus particle in NK cell culture system. Some NK cells will get transduced, and the cells will turn green and express the artificial antigen receptor on the surface, and then we can use antibody to staining to detect by flow cytometry. So here we include an untransduced image to look at the background of the untransduced NK, and here is the mucin-1, our mucin-1 CAR NK. Here you can see about more than 60% NK cells were CAR positive. So again, we use these transduced NK cells to doing cytotoxicity. Here you can see a targeting on the mucin-1 TM positive Jocata cell line. Only our mucin-1 CAR NK exerts the highest killing capacity. So here I have introduced the previous two concepts to enhance the function of NK cells. In the later chapter, I will introduce the strategies to reduce the inhibitory signaling on NK cells either by a blockade antibody or use gene knockout strategy. So we targeting on NKG2A, which is an important inhibitory signaling on NK cells, and it can bind the ligand, which is called HLA, on the tumor cells, and then it will transmit the inhibitory signaling. It's the KLRC1 gene is coding NKG2A receptor, and we use the CRISPR-Cas9 technology targeting the exosome 1. So here is the flow cytometry image. In the left panel, you can see the black line is the NKG2A wild-type group, which is high expression NKG2A in primary NK cells. But if we use the CRISPR-Cas9 knockout, the main population will going shift to the negative population. And here's the statistical data. We summarize about 20 donors, which is from about 70% NKG2A and going to about 15%. So it's a huge reduction of NKG2A expression on our primary NK cells. So again, we also conduct a killing assay. Follow the chapter 2. We want to enhance the NK cells capacity by anti-HER2 antibody. We targeting T47D, which is a breast cancer cell line. Here you can see either anti-tumor antibody, it will enhance about 50%. And if we add the anti-NKG2A blockade antibody, it will further enhance the killing capacity of our NK cells. But when we add our CRISPR-Cas9 knockout NK cells, it will further enhance the killing capacity of NK cells. So based on these results, I have several conclusions. First is if we remove the sugar chain on the Fc domain of an antibody, can further enhance the ADCC capacity in NK cells. And the second is our mucin-1 CAR NK has been successfully generated and can kill the mucin-1 TM positive cell lines. And the third, reduce the inhibitory receptor by gene knockout strategy, manifest the strong much stronger blockade efficiency than antibody blockade. Based on these observations, I have some perspective for the NK cells based cancer immunotherapy. First is the antibody can be further modified, can be further be engineered to combine with NK cells once applied into cancer immunotherapy. And the second, CAR-NK cells will becoming a promising therapy for cancer. And the third is combination therapy can help NK cells to overcome the inhibitory tumor microenvironment. At last, I would thanks to my supervised teams and my colleagues, and also the collaborator within the institute in Maastricht University, and also the international group. And also would like thank the support from CSC grant. Thank you for your attention. I will give words back to Prorector. Thank you.
Thank you very much, Mr. Candidate, for your very nice presentation, well illustrated, a little business type like, I must say, I must confess, it looks very elegant. And I think we have now plenty of time in the next 45 minutes to discuss your thesis and the outcome of the thesis. We will have 6 opponents, as you might know, and I would like to give the first opponent the opportunity to discuss the thesis with you. And I would like to give the word to Professor De Ruyser. He was also the chair of your assessment committee. Professor De Ruyser.
Thank you very much, Mr. Chairman. Thank you very much, dear candidates. First of all, I want to congratulate you, your whole team, but also your family with this fantastic and interesting and relevant work. But of course, I want to discuss with you some general aspects. And as I'm a clinician working in solid tumors, I want to first ask you, how would you overcome with your NK cells the resistance what we have due to the microenvironment? Because in general, we know that engineered cells, they are also displaying some resistance, which are very much overlapping with that of, for instance, checkpoint inhibition. So how would you tackle the microenvironment, for instance, hypoxia, accessibility of cells in the tumor, and so on?
Dear highly esteemed opponent, thank you for your valuable questions. Based on your question about how to overcome the inhibitory tumor microenvironment using our NK cells. So the tumor microenvironment is a very suppressive because they have a lot of stroma cells, and we also have like hypoxia conditions and also low pH condition in the microenvironment, and it will inhibit the effect like T cells and NK cells and also dendritic cells. As you mentioned, one approach is maybe we can use the checkpoint inhibitor because they have successfully applied in clinical and also exert potent anti-tumor capacity. And also we can use several combinations after the immunotherapy. We can also do the local radiation, and I think in our master group I have also done a lot of investigation these combinations and therapies to overcome the inhibitory microenvironment. Thank you.
So that's nice, and also nice to mention our work. But on the other hand, I still question your answer that we can use anti-PD-1 or anti-PD-L1 monoclonals for that, because we know that one of the issues, for instance, in lung cancer with resistance for anti-PD-L1 is actually that there is hypoxia, there is expression also of ligand on the tumor endothelial cells, which actually are the reason to have some resistance for anti-PD-L1. And we know, for instance, from CAR T cells, that exactly the same mechanisms play a role for resistance for CAR T cells. So I was just wondering, are you not going to introduce the same level or the same reasons for resistance in your engineered, in this case, NK cells with your antibody, and which is overlapping with the resistance for checkpoint inhibition? Is it not, let's say, a kind of generic problem what we are facing? So how would you tackle that?
Thank you for your question. So as we know, the CAR T is very advanced technology, and have already, there are 5 products now, and but basically was targeting on the hematology. For the solid tumor, is still not available CAR T products. So yeah, one approach to answer your question, we can engineering anti-PD-1 on CAR-NK or CAR T on the T cell or NK cells to directly overcome the PD-L1 inhibitory with activation signal to directly after binding with the PD-L1 ligand, then it will instantly activate the NK cells. And also we can use some cytokines to boost the function of NK cells or T cells, and we can to introduce the more T cells or NK cells infiltrate the tumor lumps, then it will can irradiate the tumor more efficiently.
Yeah, indeed. And actually, one of the reasons why thus far engineered CAR T cells fail is actually that indeed the response is much lower than expected in many solid tumors, actually. Now, I have a very short last question. Would you envisage to combine your strategy with existing drugs, I mean repurposed drugs, not the anti-PD-L1, but other drugs? Have you thought about that?
Yeah, I think so. Thank you for your question. So there's a lot of components in the microenvironment. Based on we want to conquer the cancer disease, we need several approaches to overcome the inhibitory resistance. We can use except use CAR T or CAR NK. We can also use like drugs, anti-TIGIT or based on antibodies, because it is also very specific, can only kill the tumor cells without damaging the healthy cells.
Okay, thank you very much. I'm very pleased with your answer, and I give the word to the pro-rector.
Thank you very much. And the opposition then will be continued with Professor Wang, and I would like to welcome him all the way from China. So we have a discussion between China and China in Maastricht in the middle, and I was very much looking forward to what kind of questions you would like to address to the candidate. Professor Wang, the word is yours.
Okay, first of all, congratulations to Ying. Did wonderful job during 5 years in Holland, so very good. Yeah. My first question is, you know, at the moment, for the NK therapy, usually we use NK cell derived from cord blood or peripheral blood. Would you think 2 types of the NK cell derivation, for example, derived from the cord blood and derived from the peripheral blood? Do you think there is a difference? What is the advantage? and disadvantage these two original NK cells? Can you give me some explanation?
Dear highly esteemed opponent, thank you for your valuable question and thank you for your kind words. Regarding your question is about the advantage and the disadvantage compare the cord blood and the peripheral blood NK cells. So as far as I know, the first from the percentage, cord blood can have a little bit higher percentage of NK cell portion. In cord blood, there are more than 20%. In the peripheral blood, it's about 10% or 15%. So this is one different. And the second is the phenotype or the functional in cord blood are more immature in these cells, and in the peripheral blood they are maybe already met with autologous antigens or other antigens. Then they become more mature. And then and the third is as far as I know, as the different stage of the maturation from these two sources, the peripheral blood NK cells may be exert much higher killing capacity than the cord blood. This is my answer I know. Thank you.
Thank you. Another question regarding to the CAR-NK. In your work, you use NK cell from the peripheral blood, but you know, for the MD Anderson's work for CD19 NK, they
Yes.
used the CAR-NK from the cord blood. Do you
Yes.
think, for your point of view, I know so peripheral blood maybe is a little bit difficult to transfect it. So why do you use cord blood? Or why do you?
Thank you for your question. Yeah, the Cathy Rajavani group from MD Anderson, they have conducted their first clinical trials by using CD19 CAR-NK, and their source of NK was derived from cord blood. And there are also a group using the peripheral blood. Regarding your question about the difficulty for transfection, introducing the CAR gene into these two types of NK, I think the first step, we need to activate these two types of NK in vitro, because these two types of cells were derived from human body, and in human body there's a lot, maybe different mediums of cytokines to maintenance. But when we transfer these cells into experimental lab, and we need first let them fully activated, and then these cells can be much more easily get transduced. As far as I know, both cord blood and also peripheral blood, if they are fully activated by K562 membrane-bound IL-21, IL-4-1BB at 7 days or 10 days, they can much easier get transduced. Yes, so my answer may be first, we need to use methods to fully activate these NK cells, and also second, we need a higher titer of our CAR-NK virus, because basically we always use the lentivirus or retrovirus, and the titer is important, and the NK cells are naturally resistant to viral infection. So, and also even this Monday, I read one paper published in STAR protocol, which is a cell journal. They demonstrate the peripheral blood after transduced with the third generation HER2 CAR, after 2 weeks, they also will disappear. The internal mechanism is still needed to be further investigated. Thank you for your question.
So my third question is, do you know the CAR T is very popular compared to the CAR NK?
Yeah.
Would you think for the between CAR T and CAR NK, how to make decision to select CAR T or CAR NK? What is the basic principle to make this selection?
Thank you for your question. So currently, there are, as I mentioned before, there are 5 CAR T products approved by US FDA. 4 was targeting CD19, and 1 is targeting BCMA, this antigen. So the CAR T is there's the advantage is much bigger percentage in our human blood, so it's much easier get expanded. But NK cells is much less percentage, and has much higher killing capacity towards tumor cells. So when we met this situation to choose the CAR T and the CAR NK, we would think about the disadvantage from these both 2 sides. As far as I know, the CAR T has the T cell receptor, and even if we introduce the CAR or gamma delta receptor, they are maybe much more easily get the risk graft versus host disease. This is the one risk, and the second is CAR T. They will expand in vivo, and then they will secrete a lot of cytokines, will have the risk of cytokine release syndrome. So CAR NK don't have TCR and have shorter lifespan. So currently, CAR NK is a hotter research field, I would say. Thank you for your question.
So maybe I have a last very short question.
No, I'm sorry, Professor. We do have to proceed. Sorry.
Okay, thank you. Back to the project. Thank you very much. Thank you for
Thank you very much, and sorry to apologize. We have still 4 more to go. So
Okay,
the opposition will be continued by Professor Kubal. He is professor in hematology at Utrecht Medical Center, and I would like to give him the word. Professor Kubal.
Yeah, thank you very much, and dear opponent. First of all, my congratulations to this wonderful thesis, and of course also to promoters and co-promoters, which is a lot of amazing work. And I really sort of liked going through it. It's one of the rare events that I really have such a big paper version in front of myself. And I sort of got more and more intrigued, and at page 148, at least in the version which I have, there are all these boxes, and the box 0 is titled "My Personal Road to the Generation of CAR NK Cells." And then you go and most likely it was like sort of in a lab journal through different strategies. And I just want to first get your thoughts on page 149, the box 1, on the schematic CAR NK vector maps, because this always to me looks a bit like playing Lego. Yeah, and maybe you can elaborate a little bit on your thoughts why you played Lego for construct 1 different than for construct 3 or for construct 5.
Yeah, highly esteemed opponent. Thank you for your question. So on page 146, so I think here's my version, maybe different version. I see a lot of boxes with one construct have different activation signal. This is what you mentioned. Am I correct?
Yes, and before we go into this, this looks very good indeed. So we have, I have, not sure whether you can see me, I have a little bit different, but it looks also
okay.
great. I still have the better version, yeah. And the first thing I was wondering is in terms of detection, because one of the challenges always if you express CAR-Ts is how you detect them. And you made a certain choice of a P2A element with an LNF, LNGFR, and then an E2, EF1A, and then a GFP. This is rather complicated, or?
Yes. So our lentivirus have two promoter, which so the GFP was under the EF1 alpha, and the other promoter was called CMV, and the CMV was promoter for our Fury CAR. And regarding the different boxes, this map is the work I did in our lab, and I generated all these kind of CAR NK vectors in one lentiviral plasmid vector.
Yeah,
so,
I'm impressed. That's a long list, but isn't the risk? I mean, maybe you can elaborate on risk if you use two different promoters, just for technical lab reasons. So what might be the risk?
Yes, thank you for your question. Yeah, one, as far as I can concern, there is different promoter will charge for different gene of interest. So EF1 alpha was for the GFP, and the CMV was for the CAR. The risk is they are maybe out of the range for the lentivirus, because the capability for a lentivirus is 4 kb. So we already think about, but at that time we can just simply remove the EF1 alpha, use another P2A as you suggest. Yes, this is a good suggestion.
Yeah, so for example, like it's not for T-cell receptors, because you run into the risk that the GFP doesn't necessarily reflect the CAR-T expression if you have two different promoters. And then I know another opponent is way much more interested in all these different constructs you made, but can you, out of these, pick me your favorite construct and tell me why you think it might be the favorite construct to further pursue? So I see 18 choices, but of course not all 18 are MAC1, but so the lead doesn't interest me so much, but sort of from the other ones, you have a lot of different choices.
Yes, I would like to. My favorite vector was 18. So my explanation is so at the beginning we don't we want to screen from previous 11 vector to choose one best intracellular activation and signaling for generating CAR NK, and we going to Germany, Professor Winfried Wells group, and also in Tianjin, Professor Wang's group. So this 18 was after these two labs study experience, and we optimized them, and also we have successfully generated based on the 18th lentivirus construct.
Okay, so it was a long road to come finally to 18. I'm not sure whether you intentionally made it the last one, but it's fair enough. And then on the next box, so it's really very nice and well done. So then you come to the next quest you have, which is sort of the type of vector you use for transducing. Then you have lentivirus, then you have retrovirus, then you have AAV6, and then you have in box 4 electroporation. Can you a little bit elaborate on pros and cons, and again how you found your final strategy?
Dr. Gong, can you give a short answer?
Thank you for your question. Yes, so AAV cannot transient the expression in NK cells, but can easily have high titer. And the retrovirus now in clinical trials, we don't maybe have a risk to get a leukemia risk. And lentivirus seems much more safe. And the electroporation approach is they can easily they can only maintain that maybe for 48 hours or 72 hours. After that, they will disappear. So, but the advantage of electroporation, they will not integrate in the genome, and this is also very efficient with mRNA electroporation. That's my answer.
Thank you very much. I sense that my time is finished, and I give the word back to the prorector. Thank you very much.
Thank you.
Thank you very much. And indeed, we have swiftly to proceed to the next member of the Corona, Professor Mittis. He is a member of the assessment committee and is professor in immunotherapy, and particularly for hematology oncology diseases from Amsterdam. And I would like to welcome him, and I'll give him the word. Professor Mittis.
Thank you, Prorector. And dear candidate, I would like also to congratulate you and also your promoters and co-promoters with this very nice preparative work, which finally may make NK cells competitive for T cells, which is of course the dream of your promoter. We know that for a long time. And having said that, I read your thesis with very interest, and especially Dr. Kubal already told that this chapter 4 was for me very interesting because we are also interested in CAR T cells. And when I was reading that chapter, I had a lot of questions. One of my questions is actually, you are reviewing a lot of papers and I'm saying that, okay, these CARs are generally designed for T cells, but they are also are good for NK cells. And in one of those sections, I would like to see some biology of NK cells, but you are not mentioning about biology of NK cells, if we are doing good or not. So the point is actually what I want to raise. What do you know about co-stimulation of NK cells?
Yeah, highly esteemed opponent. Thank you for your valuable question. As far as I know, for the co-stimulator signaling for NK cells, from my textbook in my college time, maybe a little bit older, but the principle is NK cells co-stimulator signal. One is based on IL-2. They need the signaling from IL-2 receptor transduction activation, and the second is from the CD3 zeta, because the CD3 zeta have 3 ITAM domain and they can activate NK cells very potent. And recently with the technology development, there are also several molecules like 2B4. And also the DAP10, DAP12 also can activate NK cells very efficiently.
Thank you. That's actually exactly what I wanted to hear, because I don't hear anything about, when you are talking about stimulation or co-stimulation, about anything CD28 or 41BB, because these are T cell specific molecules. And the question is, do we need them in the CAR NK cells?
Thank you for your question. Yeah, so the profile of T cells and the NK cells are different. Yeah, and also T cells have much more co-stimulators signaling maybe than NK, and their killing mechanism is different. So T cells through the TCR receptor, and or maybe the use through the CAR now, and NK cells directly from the cell contact, and they are based on the ligands, like in my introduction, the activation signal receptors. Once they recognize the ligands on the tumor cells, like through the fast ligand and the fast, they can introduce the apoptosis to the target cells to kill the target cells. Yeah, thank you.
It's exactly the point what I want to make is you have made 18 different constructs to study a lot of different possibilities with Lego thing, but you didn't even try a construct without a CD28 or 41BB. Why not?
We do have several factors without CD28 and the 41BB. We have first generation only CD3Z and the DAP10 and the DAP12.
And then did you test them also whether they work okay or not?
So from the literature as far as I know, the first generation only one co-stimulation signaling is maybe they are much shorter and they will much higher transduction efficiency.
Yes,
for
that's
NK
exactly
cells.
what I want to reach. Actually, you need to make the construct shorter.
Yes, so they get more transduced and they will give them more activation, and then several studies just demonstrated only the first generation work the best. And as also read recently, your paper have also have 10 CAR T cells towards multiple myeloma and with different affinity. Let's also have 41BB CD28 and yeah, from your results, I know this activation co-stimulation signaling will influence the outcome of the therapy by using CAR T or CAR-NK.
Okay, but then my final question is, you already mentioned that the NK cells are advantages because they don't live longer. But people, for example, in the MD Anderson, they want the NK cells to live longer by using IL-15 cytokine in introduction. So would you suggest that it would be a nice idea to leave out that CD28 molecule and instead of putting an IL-2 or an IL-15 molecule in the CAR?
Thank you for your question. Yes, the persistence of NK cells is a critical issue for NK cell-based immunotherapy after they are transfusion back to patient. There are several clinical trials who failed just because NK cells are not get persistent, living much longer. Regarding your question, yeah, indeed, if there are some more research have demonstrated CD28 maybe not necessary for NK cells, we can indeed replace this cassette with L15 or L2 these cytokines and further boost.
Well, yeah. Thank you. I hope you can be managing that construct. I don't have any time, I guess, or do I have one?
No, you don't have so.
Okay, thank you so much. I am very much pleased with the answers and thank you.
Thank you.
Thank you very much, right on time. And we will continue with another opponent, Professor Beckers. He is also the secretary of our defense this afternoon, member of the assessment committee and professor at hematology at our university. And I'd like to give Professor Beckers the word.
Thank you, Mr. Pro-Rector. Dear candidate, I've read your thesis with pleasure, and my congratulations to you and your supervisors for this impressive work and also your achievements in completing the studies. We have to remember that part of it was done during the lockdown phases due to the corona crisis. So in my opinion, you have succeeded in improving many steps in the development of NK cells to make them more suitable and efficient in cancer therapy. And it's not my expertise to discuss in detail the design of the experimental work. but rather I would like to put your results in a more broad perspective and give a more philosophical element to this discussion. So after reading the chapter 4, in which you have summarized the literature specifically on CAR-NK cells, and you have referred to an astonishing number of references of 238, my first and simple question would be, are we, or more personally, are you ready to produce these NK cells or modified NK cells or CAR-NK cells for cancer therapy in humans?
Dear highly esteemed opponent, thank you for your valuable question and kind of words. So I have been studying 5 years in Professor Gerhard Boss' lab, and this lab have more than 30 years experience doing the tumor immunotherapy. So recently we have generated one method based on the K562 feeder cell expansion system, and with these feeder cells we can expand the NK cells for more than 100 times or 10,000 times within 2 weeks. So we can reach the number of GMP or clinical trials qualified NK cells. So we also have successfully generated a mucin-1 CAR-NK based on our experience, and I think with this experience we can easily transfer this research output into clinical trial towards like breast cancer or multiple myeloma. That's my question.
That was my next question. Which cancer would you prefer to treat first if you were able to produce enough quantities of cells? Because there are many questions still has to be answered. And sometimes I got a feeling while reading the chapter and all the improvements that because you are in the lead and being ahead of things, it might also causing slowing down the process. For instance, the time that these modified cells are available, still newer and more innovative ones are there. And should we wait for newer ones and more efficient ones, or what is your opinion on that?
Sorry, can you repeat your question again? Sorry.
Now, there's this improvement which you have described in your chapter, and that are in your review still newer products are on the market or being improved.
Yes.
And you have done your part in improving the CAR-NKs and NK cells. So I got an opinion that because these improvements are so rapid that we still have to wait for more improvements before we could use them in humans.
Thank you for your question. I think in clinical trials, there are very malignant disease like cancer. These patients have time limited. So and we also very critical think about which regimen is best for cancer immunotherapy. And there are also several cell products like based on the T cells. We have CAR-T and gamma delta T and NKT for NK cell.
But going back to your work, because you have produced, you've done your utmost best to improve the NK cells or the CAR-NKs even. And in chapter 1, you state yourself there's an urgent need to develop novel cancer therapies, and you point specifically at cell immunotherapy. So again, the question is, are we there yet, or do we have to wait for better cellular options?
Thank you for your question. Yes, so the cancer immunotherapy is a very fast development field. Currently, the clinical trials of first CAR-NK, they have exist encouraging an outcome for the CD19 and CAR-T, but we are still a lot of cancers lack the suitable target and suitable treatment. I think we still need several steps to get the final conclusion.
Last question, if I may, Pro-Rector, because I still looked at your experimental design work, and you had as outcome measure the cytotoxicity effects. You showed some pictures of it. Do you think, because I saw an effect and a target ratio, the amount of NK cells is of value in predicting the effect? Do you think that some kind of test could predict the efficacy of the treatment in humans?
Thank you for your question. As we know, cancer is very heterogeneous, and also patients are virulent. Based on also from the clinical experience, we can follow like use the drugs, or we can estimate how many NK cells we need to cure a specific cancer. But maybe the patient is virulent. We should think about some other parameters to take into account. That's my answer. Thank you.
And just another point.
Sorry, Professor Beckers. We have to continue with our last opponent before we closing. Thank you very much. And that's why I would like to give the word to Dr. Dubois. He was also a member of your committee, and he is affiliated with the Department of Precision Medicine. And I give you the possibility to discuss with the candidate.
Thank you, Mr. Pro-Rector. So, dear candidates, also I want to congratulate you and your promotion team with this splendid work. I really enjoyed reading your thesis. And I immediately want to go to chapter 5 with you, because immune checkpoint inhibitors nowadays receive a lot of attention. A lot of clinical trials are ongoing. A lot of them are currently approved by FDA and EMA. So that's why I found the NKG2A also a new interesting approach. But I'm a bit confused. If we go to figure 2 on page 165, you use the 3 multiple myeloma cell lines, one which is HLA-E negative, and the other ones are both are positive. And if you incubate those with NK cells, you see a nice degranulation for the ones who express HLA-E, and no degranulation for the negative one. But if you then look at 2C, your percentage tumor cell kill shows kind of a different pattern. So where you don't have HLA-E, suddenly you see upon NK incubation a reduced tumor cell killing. So why, or can you explain me why this dichotomy between degranulation and reduced tumor cell kill?
Esteemed opponent, thank you for your valuable question. Regarding your question is about the relation between degranulation and the cytotoxicity. So the mechanism of NK cells killing a tumor is by secrete the granules. The granule is very important killing weapon from NK cells. But also maybe there are also some other mechanism can also induce the target cells apoptosis. We have several assays also with antibody. We found that if we incubate the NK cells and the tumor cells even present the antibody, we see the degranulation is not always correlated with the killing. Sometimes the degranulation will much more, but actually the killing is not that good. That's
And
my
what
answer.
other mechanisms might play a role there then?
Yeah, and I think so for NK cells killing mechanism, one is based on the cell contact. They can use the fast ligand binding with the fast on the tumor cells. This is one mechanism, and also there was some cytokines like interferon gamma and the TNF alpha can secrete by NK cells can also kill targeted cells. That's my understanding. Yes, thank you.
Right, thank you. And I want to move back to also chapter 4, because in table 7, you mentioned there are a lot of interesting targets with the common ones, EGFR, HER2, PSMA. And in your acknowledgements, you acknowledge Professor Debets for the CA9 CAR virus. Now, CA9, I've been working with CA9 for 10 years, so I'm actually interested. Why do you think CA9 would be a good target to make CAR NK cells from? Sorry.
Thank you for your question. Yes, we have contacted Professor Reno Debets from Notre Dame to ask some virus supernating. We want to generate a CAR in lab. At that moment, we don't have a positive control. So they already made a virus particle, and we want to simply get some and to make a positive control for staining and for the later experiment. Thank you.
But why do you think CA9 would be an interesting target?
CA9 is so Professor Reno Debes already conducted clinical trials by using CA9 CAR-T from I think 15 years ago, and they can kill the tumor cells in vitro, but when these cells infusion back to patient, they have very severe side effects. They can kill some liver cells, so that's why they also stop their clinical trial.
Okay, because CA9 is an endogenous hypoxia marker.
Well, as you notice, the time is over, but Dr. Dubois, I give you the opportunity to finish your question short, and Dr. Gong to answer short. Yeah, if you want, of course.
Very, very short. C9 is an endogenous hypoxia marker. Do you think that CAR NK cells have a future in targeting hypoxia?
Thank you for your question. Yes, CA9, as the first professor mentioned, the tumor microenvironment, like the CA9 will also highly expressed in the tumor microenvironment. It can be an interesting target for CAR NK and CAR T. In the later experiment, we can use the CA9 as a good target, but we need to think about the side effects, the off-target effects. Thank you.
Thank you very much to all of you. Thank you, Mr. Ying Gong. The time appointed for defending your thesis is now over. The degree committee will now go back to their breakout room, will withdraw, and we're going to discuss the quality of your thesis, but also the way you have defended your thesis this afternoon, or for you this evening. I request that you wait with your family and friends here in this room till our results and our deliberations are over, and we will return in about 10 minutes. Thank you very much. And the office, please bring us back to the other room.
Thank you.
Mr. Prorector.
I'm not quite sure if we have the right figure. Joshua or Luke, is everything all right?
According to my list, it's okay.
Because I have a screen now, which is a black PhD office. I don't have a full screen with the candidate.
That is Luke's camera.
Okay,
So
good.
we have all the members are here.
So can you bring us back to the aula?
We are.
I don't see the candidate. Okay, maybe I should use a different screen here myself. Okay, I switched myself. Very well. Mr. Ying Gong, the degree committee here present online has assessed the quality of your thesis, and in particular the way you have defended your thesis this afternoon or your night, your evening in China. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Germeraat, or Dr. Germeraat, is authorized to confer you this academic distinction in accordance with Dutch university customs, and I invite your supervisor now to take the floor and handle the official matters. Dr. Germeraat, I give you the word.
Thank you, Mr. Prorector. Dear Mr. Gong, first I would like to ask you a question.
You can sit down, Dr. Gong. It's easier. Okay, thank you.
Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible?
Yes, I do.
Then, by the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer you upon Gong, the doctor, and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and your supervisors, affixed with the official seal of the university. And this will be shown by the beadle.
Thank you.
Dear Dr. Gong, dear Ying, it's my pleasure to congratulate you as first. Happy? Congratulations. A job well done. I would like to give the word to Dr. Roel Kleinwoltering, your co-promoter.
Thank you.
Thank you. Very learned Dr. Gong, dear Ying. Must be a little strange sitting in front of your computer in China to realize that you've now completed this last step of a journey that started 5 years ago. And even though you had been to Europe before, you had never been to Maastricht before. Now you're back in your home country in a multi-million vibrant metropolis with memories of this small city in the Netherlands. Memories of which some will stay with you for the rest of your life. Your PhD started after a couple of meetings online, and you took a plane from China, brought with you a big suitcase, and arrived at the train station in Maastricht. Gerard picked you up and drove you to your new apartment, only to find out that there wasn't even a bed. And I guess that was an interesting way to get to know your supervisors, Wilfred and Gerard, that would always try to help you out, not only scientifically, but also with other matters that occurred. After a false start, you quickly set off an ambitious plan of making CAR-NK cells. And we had already discussed a strategy for that, the Lego block, so a comparison of 11 combinations of signaling molecules. It turned out to be much harder than we all imagined. In fact, you worked on this part for the full 5 years of your PhD. And even though we haven't generated the ultimate CAR-NK cell yet that is ready to go to the clinic, this journey turned out to be a very productive and interesting one. You very nicely described everything that you tested and learned in an extensive chapter in your thesis. And we published this as a review in a high impact journal, already resulting in PhD students contacting now because of this publication. And at the same time, it was an important test of your motivation and perseverance. We never had to worry about your determination to get the project done. You worked hard, very hard. You worked a lot. And when people showed up in the lab at odd hours, you would always be there. You worked so fast, resulting in huge piles of data. And in the beginning of your PhD, you would overwhelm everybody with presentations that contained at least 25 graphs per slide. And often you would find ways to fit even more plots on a single slide. For me, your ninth proposition plays a central role in guiding you as a PhD student. Less is more. Wilfred once told me that your experiments and your presentations were as expensive as the Chinese Great Wall. I would think that if someone compares your scientific work to this impressive construction, that's not necessarily a bad thing. But even so, we kept repeating that less is more. And while you importantly improved your academic English, you also started telling your research as a story. Your presentations and papers would have a clear introduction, a question to be answered, still a huge amount of data, and then a conclusion. You developed several projects cited as side projects into full papers that we published over the last few years. And these projects required you to establish new techniques in the lab that are now routinely applied in other projects. In your thesis, you describe CAR-NK cell development in the lab as an experimental road that you took to generate CAR-NK cells, with all the bumps and obstacles that you met along the way. And the people that worked with you are all aware that there were many hurdles along the way. And even though that journey isn't finished yet, you're sharing your knowledge that you obtained as an interesting set of experiments in your thesis. And this statement about the bumpy experimental road nicely illustrates how you worked in the lab. You worked on your own project, but at the same time, still cared for all the other people in the lab. You're a very good team player. You were available for other people to answer their questions. And it was never a problem to pass their cell culture or to analyze some more cells on the flow cytometer. You shared your protocols, collaborated on a number of projects, and you were involved in the supervision of various students here in the lab. You guided them on their path by making them a new member of the team and by giving them their own project and responsibilities, while you also kept a close eye on them. Your road to the generation of CAR-NK cells also took you to collaborations here in-house and abroad. You closely worked together with the people in the Mastro lab, especially with Arjan de Groot. Together with Wilfred, you paid a visit to a German collaborator, and with Gerard, you went to the laboratory of Professor Wang in Jiangyin. On these occasions, you quickly engaged in scientific discussions and were able to initiate new collaborations. This is an excellent demonstration of your scientific expertise, but to me, it mainly illustrates the important skill of scientific collaboration that you master very well. During your trip to China with Gerard, you took the opportunity to guide him around some highlights in China. You showed him the Forbidden City and the Great Wall. And in your acknowledgement, you cite a poem from Mao Zedong, who said that one who fails to reach the Great Wall is not a hero. An interpretation of this saying would be that all great people must overcome difficulties. And as you said, your road of improving CAR-NK cells for cancer immunotherapy was windy and certainly bumpy. Today, we concluded that your journey was well worth it and made you a wiser person. I know I can speak for the three of us as your promoters when I say that we're looking forward to keep collaborating with you. As from today, we're very glad that you decided 5 years ago to join the lab in Maastricht, and we got to know you as a very bright scientist and the friendly, warm person that you are. Your country must be very happy to have you back, even if it would only be for a number of years, to care for patients and to further shape your scientific career. I wish you and your family and your friends a happy celebration of this major achievement today and all the best for the future. And with these words, I'd like to give back the floor to the program.
Thank you.
Thank you very much, Mr. Klein Woltering. And of course, I first would like to congratulate the new young doctor. So Dr. Gong, your body language, even in about 10,000 kilometers away from Maastricht, is pretty clear. You're pretty pleased, and I would like to congratulate you on behalf of the board of Maastricht University. I would like, in my congratulation, I also put forward your family and your friends who will join this online defense. Of course, It would even be better if it would have been done in our aula here in Maastricht, but the circumstances are differences. I also would like to congratulate, of course, your team who has brought you to this event in these last 5 years, and I've seen you growing in these last 5 years as well. I also would like to thank the members of the Corona who joined us this afternoon, who prepared questions and who took time in reading your thesis, and I really like to congratulate you. I would like to end with something personal to you, of course. You have an important task, not only going back to China, not going to work there, but now you from Maastricht University, one of our ambassadors over there, because it is our aim to bring a lot of European, Dutch, German, Belgian students to China. And one of the labs in which we like to send students will be your lab later in China in the future. You are one of the 282 CSC students who have worked in Maastricht and who have started to graduate. And basically on our list of CSC students, you are number 63 on the list who finished their CSC PhD thesis in Maastricht. So I wish you a very successful career in your event. I understand from Dr. Hermrath and Professor Bos that they will continue working with you, guiding with you. And indeed, we hope there will be opportunity after the pandemic to visit, or that you will return here in Maastricht. Saying this, I would like to declare this thesis defense officially for ended, and I wish all of you a happy day. Thank you very much.
Thank you.